and welcome to CBD Journal Club, presented by our sponsor, CBD Health Collection. And today we're going to be talking about a study. The name of the study is Oxidative Stress and its consequences in the blood of rats irradiated with UV, protective effect of CBD. Let's get some background terms here first. Oxidative stress is something we've talked about on the podcast, but if you haven't listened to those episodes, what this is basically an imbalance between our antioxidants and things called ROS or RNS, reactive oxygen species or reactive nitrogen species. What these are molecules that damage our proteins, damage our fats, damage our cells, damage our DNA. So that's bad. If we have more ROS or RNS than we have antioxidants, this overall leads to damage in our body. And then irradiation. When we think of radiation, we think of glowing green men, right? But actually what radiation is a transfer of energy from particles or electromagnetic waves. And in this regard, we're talking about light. Light is a particle. So light energy in the form of UV. And we know about UV. We're going to move into the introduction here for the study. UV radiation reaches the Earth from the sun, and this is split into two types. There's UVA and UVB. The difference between these is how deeply they penetrate. It's related to the energy of the spectrums. UVB penetrates the epidermis, which is the top layer of the skin, affecting metabolism of the cells. UVA penetrates much deeper to the level of the dermis, and it actually influences metabolism of fibroblasts, and fibroblasts make growth factors and healing agents for the skin. They're very important in wound healing. Also infects dendritic cells, which are a type of immune cell, and other immune cells as well. Now UVA also reaches our microvessels, our capillaries, which bring blood to the skin, and this can cause system-wide circulatory effects because you're able to reach the circulation. Prolonged exposure to UVA and UVB increases reactive oxygen species and reactive nitrogen species, which are the two most dominant types of particles in our air. They're also the two most dominant things when we talk about oxidative stress. And this is caused by inducing something called NF-κB. We've talked about NF-κB on the podcast before. It is the master switch for pro-inflammation, and it reduces NRF2. NRF2, we've talked about, is the master switch for anti-inflammatory molecules. So usually there's a balance between these two, between promoting inflammation and reducing inflammation. Now the overall result of UVA and UVB excess exposure, we're not talking about the usual 15-30 minutes of sun exposure that's healthy for us. We're talking about excess exposure results in more oxidative stress. This is why sun damage happens to our skin. It results in more lipid peroxidation. We've talked about lipid peroxidation before on the podcast. Basically, these are lipids that have been oxidized. They've been attacked by these reactive oxygen and reactive nitrogen species, and these cause a lot of inflammation. In fact, oxidized lipids are what causes heart disease, atherosclerosis, strokes, all that bad stuff. Excess exposure to UVA and UVB light contributes to skin cancer and various skin lesions. In fact, it's estimated that 60 to 70% of malignant melanoma is caused by UV exposure. Now, why are they looking at CBD with this stuff? CBD has antioxidant properties when topically applied. I actually used CBD Health Collection lotion for the reasons we're going to be talking about in this study. Now, this is a rat study. You can't generalize it to humans, but the science supports using topical CBD for antioxidant properties. One study found that applying CBD following UVB exposure causes CBD to accumulate in cells. It then balances the activity of pro-oxidant and antioxidant compounds. This is what we've talked about so many times before. CBD works as a balancing agent. and it regulates the production of these reactive oxygen species, and it also regulates the production of the major pro-inflammatory cytokines. Cytokines are messengers. They're how our cells talk to each other, and that's TNF alpha. And it balances NRF2, the anti-inflammatory signal, with NFKB, the inflammatory signal. It also has been shown to modulate lipid metabolism in the skin. Preclinical studies, these are not clinical trials where there's a placebo group, but they're basically proof of concept studies. These studies indicate CBD may be effective and supportive for eczema, psoriasis, pruritus, which is itching, and inflammation. And topical application of CBD ointment improves hydration and elasticity in patients with psoriasis and atopic dermatitis. And topical CBD has actually been used in peripheral neuropathy for people who have peripheral nerve injuries and problems because of that. Now, there's no data on topical CBD looking at what we call redox balance, which is basically that oxidative stress, that balance between the antioxidants and the pro-oxidants, those reactive oxygen, reactive nitrogen species, and metabolic disturbances in the skin. This study was done to evaluate metabolic disturbances in the blood of nude rats, basically rats that have been shaved, and irradiated with UVA and UVB, and then look at what the effect of CBD was. They applied CBD topically every 12 hours, and then they measured ROS levels. They measured antioxidants directly and indirectly. They looked at measures of oxidative stress on lipid metabolism using lipid peroxides. And they looked at receptors for the ROS and TNF alpha. Let's move on to the methods portion of the study. The rats had a 12-12 light versus dark cycles, and they had 4 groups: UVA alone, UVA plus CBD, UVB, UVB plus CBD. And they applied 31 to 36 milligrams of CBD to the back of the rats, which was 120 milligrams per kilogram of body weight. This is a much higher dose than what you'd see in humans. They measured various proteins, electrolytes, enzymes, and other things like CBD levels, the endogenous cannabinoids, anandamide and 2-AG. They measured these reactive oxygen species, antioxidants. They measured enzymes involved in this process: superoxide dismutase, glutathione peroxidase. Superoxide dismutase gets rid of hydrogen peroxide-like molecules. Glutathione peroxidase and glutathione reductase are enzymes involved in the synthesis of glutathione, which is our major antioxidant. We've talked about glutathione on the podcast before. It is very important for detox, very important for our overall function. Thioredoxin is another endogenous, meaning our body makes it, antioxidant. So they measured the enzyme responsible for making that, thioredoxin reductase. They also looked at glutathione and thioredoxin, the actual antioxidant itself. They also wanted to look at antioxidants you could get from nutrition, not antioxidants that the body makes, to see the impact of these. So they measured vitamin C, vitamin A, vitamin E. They also measured protein modifications that happen with oxidative stress, lipid peroxides. These are fats that have been oxidized. And they also measured the levels of proteins in what we call PMNs, polymorpho nucleosides. These things are white blood cells, are infection fighters. So the results: CBD penetrated the skin to the blood and was taken up into the white blood cells. White blood cells have receptors for CBD. We're going to talk about that in a minute. After exposure to UVB, the CBD dropped in the blood and increased in the white blood cells. So it moved from an area of the blood to the cells where it actually caused some things to happen. It decreased reactive oxygen species generation in the blood of UVB treated rats. UVA rats with CBD, they had an increased thioredoxin level, meaning they had an increase of this self-made antioxidant. They also had increased glutathione reductase activity, meaning that they were more efficient at generating glutathione. UVB light actually had higher glutathione levels and higher thioredoxin reductase levels in the plasma. Meaning overall, what this means is the CBD increased our body's own natural antioxidant response. Now, they looked at why did this happen? Levels of superoxide dismutase, the thing that deals with hydrogen peroxide, were not increase. Levels of another glutathione enzyme, glutathione peroxidase, did not change. And there was no significant change in vitamin A, E, and C levels, those antioxidants we get from food. But there was a small increase in these. It wasn't significant, but there was a small increase. Meaning overall what this shows is that CBD caused less consumption of other antioxidants in our nutrition plan, and it increased our body's ability to make its own antioxidants. And the author suggested the result is due to a change in this redox due to changes in the balance between NRF2 and NFKB in white blood cells and changes in lipid metabolism. UVA, we know, increases NFKB activity, and while it increased TNF alpha activity as well, UVA and UVB, CBD decreased TNF alpha and NFKB in controls and the UVA rats. It wasn't affected in the UVB rats. So we're seeing that the different spectrums of light are causing different things. Now, when we go outside and we spend 2 hours in the sun and get burned, we're getting exposed to both UVA and UVB light. UV radiation nor CBD directly affected NRF2. So how did it actually work on NRF2? We're going to get to that in a minute. UV radiation increased oxidative stress, and there was an increase in oxidized lipids, those lipid peroxides, and other reactive oxygen species dependent products. There's a protein called 4-HNE, which is a downstream byproduct of high levels of ROS, high levels of oxidation. This means that proteins are being changed and damaged by the ROS induced by UV light. And CBD decreased both oxidative stress products, the lipid peroxides and the 4-HNE. UVA and UVB increase anandamide, which is important. That's one of the natural CBD molecules. And so what the body does is then it's trying to upregulate these natural things, but it decreased another type of natural endocannabinoid, 2-AG. So it was consuming that, and the body was trying to respond by increasing anandamide. An application of CBD actually increased 2-AG to the levels of the controls, meaning it wasn't being consumed to deal with the stress and the impact of the UV light. CB1 receptors decreased by 20%. With radiation, receptors are the key, right? It's what these things bind to and cause action. CBD decreased that by 40%. Now that sounds weird, but we're going to get to that in a minute in the discussion. CB2 receptors increased by 50% with CBD application in the control rats, and UVA and CBD reduced this by 30%. We're going to talk about why that's important in a minute. There's another receptor called PPAR gamma, which was increased by 50% in groups treated by CBD. We're going to talk about why that's important. But spoiler, that's where the NRF2 effect comes in. I know this is very technical. We're going to summarize things pretty much at the end. Now we're into the discussion portion of the study. UVA penetrates deep into the dermis and can affect blood vessels and lymph vessels. Lymph is our other transport system. It's one of the ways we detoxify. UVB can stimulate skin cells to release inflammatory molecules into the blood with overexposure. Again, we're not talking about 15-30 minutes of sun exposure. That can be good for us, depending on your skin pigment. Both can increase ROS and oxidative stress and damage DNA, proteins, and lipids. Chronic exposure, especially to UVB, can lead to oxidative stress, increased reactive oxygen species in the skin and in the blood. So we're talking about stuff that happens in our skin, which is our largest organ in the body, affecting the entire body. UVA and UVB stimulates NFKB to cause pro-inflammatory cytokines, pro-inflammatory messengers, and increase ROS. Remember, ROS, reactive oxygen species. This damages our proteins, damages our DNA, damages our fats, and this causes inflammation. It also modifies and decreases the efficacy of our antioxidant enzymes. So it puts our natural systems that deal with this at a disadvantage. And this puts more stress on things that our body can't make, like vitamin A, vitamin C, and vitamin E. So if you're depleted in these nutrients, then you're really in trouble. Previous evidence in mice has shown that UVA decreased glutathione reductase and catalase. Catalase is another one of these enzymes that deals with reactive oxygen species and levels of glutathione in the skin, blood, and liver. And UVB reduces superoxide dismutase, glutathione peroxidase, and glutathione. What this overall means is that in mice, exposure to these, it causes a consumption of our natural antioxidant ability. It decreases it. This leads to changes in levels of anandamide and 2-AG and downregulation of the receptors for them in white blood cells after radiation. This study confirmed that CBD, when topically applied in rats, penetrates into the bloodstream and is present in white blood cells. After exposure to CBD, the CBD goes into the cells from the blood. And CBD topically reduces ROS in the blood of rats exposed to UVB. CBD increases the synthesis of 2-AG, a natural, what we call endocannabinoid, which downregulates CB1 receptors. And this likely means that there's less ROS and TNF-alpha generation. When these CB1 receptors go up, it means that the body is responding and trying to bring back balance. And it's trying to do so by increasing this to increase the anti-inflammatory effect. But by CBD reducing ROS, reducing TNF-alpha, that inflammatory messenger, it makes it so that you don't need to increase the CB1 receptors. And then upregulation of the CB2, which these are the two different cannabinoid receptors, indicates also downregulation of ROS production and a shift towards antioxidant abundance and not ROS abundance. We talked about this is the redox. This is the balance between anti-inflammatory, antioxidant, and pro-inflammatory, pro-oxidant. And so oxidative stress, again, is creating molecules that are going to damage our body. And antioxidants, their literal job is to detoxify these things and make it so they're not damaging. PPAR gamma, that receptor that is increased, it has anti-inflammatory and antioxidant effects, because when this receptor is activated, it works synergistically with NRF2, and then it also causes downstream of this, like flowing down a river, it will inhibit NFKB. This study did not show a direct action on NRF2, so the action on NRF2 was indirect by stimulating PPAR gamma and synergizing with NRF2. And this has been shown because in other studies, CBD increases several NRF2 products, and these NRF2 products are our body's own natural antioxidant systems like glutathione and like thioredoxin. CBD causes more NFKB to be removed from the system. It causes it to be recycled, so it's removing that inflammation signal. CBD prevented lipid peroxides and other oxidative stress proteins from forming. This is good, meaning that there was less ability for the UVA and UVB light to cause damage to cells. And this effect can be enhanced by using CBD with other things like flavonoids, which are antioxidant molecules, or other whole plant extracts. We've talked about before the entourage effect, how using a full spectrum CBD, you get CBD, you get flavonoids, you get terpenes, and all of these molecules work together to augment the effect of the CBD. In fact, flavonoids have been shown to increase CBD absorption through the skin, and when taken orally, increase absorption into the brain, bypassing the blood-brain barrier. And this is why we're a fan for getting the most out of your CBD using a full spectrum CBD product. And like we said earlier, to protect our skin, we use the CBD Health Collection lotion daily. Okay, I know this was a technical study. Again, 30,000 foot view of this was this study in rats showed that CBD helped protect the skin and the whole rat body by decreasing in the blood damaging products that are made when you are overexposed to the harmful effects of sunlight. Okay, guys, well, I hope this was informative. I know it was super sciencey, but it's really cool that we're getting all these studies on CBD and its effects and how it really is this balancing agent to bring us back to our normal functioning. Thank you guys for listening. We'll be doing another video next month on a new CBD study. God bless.